Hello, and welcome to this Nature.com custom webcast titled History of Digital PCR, Revolution in Genetic Testing and Analysis. My name is Jayshan Karpan, and I will be your moderator. Today's webcast is sponsored by Stilla Technologies. We'll begin the webcast with a presentation from Dr. Wim Tripstein of Ghent University and then end with a Q&A session. To ask a question, just type it in where it says type your question here and then press submit at any point during the webcast and we will answer them today. And now over to Wim.
Hello everyone, also from my behalf a warm welcome and thank you for joining this webinar on digital PCR. This webinar will cover following aspects, starting with an historic overview and basic principles, after which I will focus on how quantification is performed, go over some digital PCR platforms, applications and future directions. So let's kick this off by visiting some landmark discoveries which led up to the development of digital PCR. So if we talk about digital PCR, we really talk about the quantification of nucleic acids. And it was Friedrich Miescher who first isolated these molecules from white blood cell nuclei and called it nuclein. This work was followed up by Albert Kossel, who further purified these substances, discovered acidic properties, and isolated the 5 nucleobases we know today as nucleotides. In 1889, Richard Altman coined the term nucleic acids, and after these purification efforts, there was a period of really figuring out what their function was. And it was in 1944 that the work performed by Avery and colleagues showed that DNA was the basis for inheritance via their famous transforming principle experiment. This work was quickly followed by the discovery of the double helix structure of DNA by Watson and Crick, also describing the base pairing of G to C and A to T. The next crucial discovery was the isolation of a thermostable DNA polymerase by Thomas Brock from the Yellowstone ponds, who took center stage in the polymerase chain reaction, which was invented by the belated Kary Mullis, who actually passed away last month during the construction of this webinar, unfortunately. This method, and I quote, is directed to a process for amplifying any desired specific nucleic acid sequence. His PCR method needs several components to work, such as primers, dNTPs, DNA, buffer, and a polymerase. After a first denaturation step, primers can anneal to the target sequence, after which a DNA polymerase binds and performs the elongation to get to the full DNA product. This process is repeated, or cycled as we call it, to exponentially amplify the sequence. After this reaction, the PCR products can be visualized on gel, at that time by ethidium bromide staining, as was shown by Kary Mullis himself in 1988 for several different products with different lengths. After this groundbreaking invention, the next question really was, how can we actually quantify this and trace back the number of molecules present in the sample of interest? Well, if we take PCR as a common ancestor, there were 2 strategies put forward in the early 90s. Digital PCR and a quantitative PCR principle. If we first look at qPCR, here a fluorescent marker in the form of an intercalating dye or probe is used to measure the buildup of DNA during these PCR cycles in real time. Next, you can use a standard curve where the fluorescence is measured of known quantities of DNA to get back to the amount of molecules that was present in the sample of interest. On the other hand, if we look at the digital PCR principle, we start from the same sample prep as for qPCR, but first we perform a limiting dilution of the sample in different partitions. Before a PCR reaction is run, and instead of following the fluorescent buildup, the fluorescence is measured once at the end of the reaction, which is called endpoint detection. Now we can actually count the positive reactions and get back to the number of molecules in the sample. Later in this webinar, I will come back to this in more detail. But for now, it's good to realize that the limiting dilution is performed where only one target molecule is present per partition before a PCR is run and there is a detection. So here you already see one of the biggest differences between these 2 principles. In digital PCR, there is no need for a standard curve and actually offers a more direct way of performing absolute quantification. However, it was qPCR, described in 1992, that was first commercialized with the light cycler and the ABI systems that came out in 1996. Later in the 2000s, the black hole quenchers were invented, which really boosted this technology. After this, several other qPCR platforms were developed, and qPCR quickly integrated in molecular labs around the world, and by now has over more than 100 FDA-approved tests in the clinic. If you compare this with digital PCR, we see that the first description of a single molecule dilution PCR was actually described before qPCR, where Riwano et al. Wanted to discriminate paternal and maternal alleles by performing a dilution to the point of one molecule as input for a PCR and sequencing. In their experiment, they ran 10 replicates of the single molecule dilution and were able to discriminate 3 alleles, as they were sure that in these replicates only one starting molecule was present. Next, Vogelstein et al., who actually introduced the term digital PCR, performed a similar dilution, but now for the detection of rare mutations of KRAS. Here they ran PCR reactions and sequencing in a 384 well plate and were able to detect 4% of KRAS mutants. They were also the first to describe how quantification was performed by using the Poisson distribution, where I will get back to in the following section of this webinar. So after this landmark paper, it took until 2006 for the first commercial platform to hit the market, and until 2011, where we really see an explosion of digital PCR platform providers. This resulted in the first FDA-approved test in 2019 to monitor chronic myeloid leukemia treatment response. So why was there a 10-year gap, and did it take so long as compared to qPCR? Well, qPCR knew a successful commercialization due to the easier setup and won the race, simply as that. On the other hand, the limiting dilution strategy was cumbersome, labor-intensive, and the technology was simply not ready. Until progress was made in the field of microfluidics and helped out to automate the limiting dilution step and the partitioning of the sample to a nano and even picoliter PCR reaction scale. This really sparked a renaissance, so you will, of this technology, and main strategies included an array-based, a droplet-based, or a combination of those systems, such as the Naica platform for Stilla's technologies. Another question is, why do we observe this explosion of digital PCR platforms as we have the well-established qPCR? Well, this is linked to the inherent benefits of the digital setup and limits of the qPCR technology. Over the past decade, several groups, such as our own, have explored these platforms and compared them to qPCR on several technical aspects. So the key features are: first of all, there is absolute quantification without a standard curve. As we know, constructing these standard curves is tricky, difficult to remake or compare even with other labs. So this really improves the reproducibility of the system. Next to this, there are reports that digital PCR is less refractory to PCR inhibitors, shows a higher robustness to primer-probe mismatches, and has a greater precision. If it comes to terms of sensitivity, there is some debate, so I will leave this in the middle, but what I can say is that with digital PCR, you will be able to pick up minority variants better than with qPCR. On the other hand, digital PCR does have a 1 to 2 log smaller dynamic range as compared to qPCR. So in conclusion, after this first decade of commercial platforms, digital PCR has the potential to become a well-established technology in molecular labs, as well as there is a growing interest in this technology. Okay, now let's zoom in on how we get from partitions to quantification, as this is the core of digital PCR. During the historic overview, I mentioned that partitioning the sample in several containers, you can actually count the molecules by counting the positive partitions. This is typically done by plotting the fluorescence of these partitions and setting a threshold above which partitions are called positive. In an ideal world, where there is an even distribution of molecules, this would look something like this. 100 molecules are distributed in 100 partitions, and you simply count them. However, partitioning molecules really follow a random distribution, and you can then end up with partitions containing more than one target molecule. So in the end, you cannot distinguish this, and you would falsely count, for instance, 63 molecules. Luckily, a famous mathematician called Poisson figured this problem out, and by using his formulas, you can get back to the amount of molecules in the sample, which, as you will see, is still based on only the number of positive and total partitions. First, you calculate the average number per partitions, called the lambda, by taking the negative of the natural log of 1 minus a fraction of positive over total. So don't be scared, it's only the positive over the total you actually need. Next, you multiply this with the total number of partitions to get back to the initial amount of molecules. If you do this in this example, you reach 99, which is a fair estimation, and as this remains an estimation, as it is not actually direct counting, You also have a confidence interval that is calculated with it. So as I said, instead of direct counting, where you have 600 positive partitions, meaning 600 molecules, it is actually not linear, but it follows a Poisson distribution, which is correct for the random distribution. This correction gets bigger as there are more positive partitions, as also the chance of getting one or more target molecule per partition increases. So this means that, for instance, 6,000 positive partitions actually represent around 9,000 molecules if you consider 10,000 total partitions. So to conclude, and if I may stress this once more, in the end you only need the number of positive and total partitions to get to your quantification. Quite elegant, wouldn't you say? Okay, let's have a look at the commercial platforms that are available. These can be categorized in three archetypes. You have prefabricated array-based systems, where the sample is partitioned via microchannels into small pockets. Next, you have droplet-based systems, where the partitioning happens via the generation of droplets, hence the name, which are then analyzed individually, like in a flow cytometry setup. Finally, you have a combination of these strategies in the form of arrayed droplets, where droplets are generated and aligned in a 2D structure. So, examples of array-based platforms are the Biomark HD, the QuantStudio 3D, the Constellation, bought by Kaizen, and the Claret. Examples of droplet-based systems are the Bio-Rad QX200 and the Raindance Raindrop, which is also acquired by Bio-Rad. Finally, you have Stilla Technologies with the Naica platform, which uses arrayed droplets and refer to it as crystal digital PCR. If you want to know more about the specifications of these platforms, I can advise the paper mentioned on the slide or refer you to the company website. However, in this webinar, I really would like to focus a bit more on the Stilla instrument. So, the same steps come back as previously shown with sample preparation, followed by, in this case, partitioning of droplets in a 2D crystal, PCR, and then fluorescence detection is performed with the possibility of data visualization. As you can see, the Naica platform offers a 3-color detection where you can visualize 3 fluorescent markers and analyze your data in 3D. So this means that you can actually multiplex with 3 different assays, which is a great advantage of this system. Another advantage of this system is the limited hands-on time, because you load your sample onto the Sapphire chip in a small amount of time, then you place it in a geode for partitioning and PCR cycling, which takes about 2 hours 15 minutes. Next, you transfer your chip to the Prism for detection and analyze your data via the Crystal Miner software. In the end, you can get to your result in around 2.5 hours, which is quite quick. If you have a more detailed look at the chip, here we have some more specifications. And if you want to compare platforms, it's always interesting to notice some of the specifications, such as the number of partitions, the input volume, and the number of samples you can run. For instance, now you can only load 3 chips in one run, making it 12 samples in total, so there are 4 samples per chip. However, a new chip will be made available where you can go up to 48 samples in a single run, increasing the throughput. This was indeed a bit of a disadvantage of this system. Recently, we acquired this system, and further in the webinar, I will show some data we generated in the context of HIV. This brings us to the applications of digital PCR. In general, digital PCR is mostly used for absolute quantification, as pointed out, but also for copy number variation and rare mutation detection. Next to this, there's a new application being developed to evaluate CRISPR-Cas9 efficiency via drop-off assays, which I won't go into now, but it will be interesting to follow this up if you're interested in CRISPR work. So if you look at these applications, you notice it is mainly DNA-based, and although RNA-based assays are on the rise, they're being used to a lesser extent. The reason for this is probably that gene expression analysis is still easily performed by the qPCR, for instance. The current major fields of interest are oncology, infectious diseases, the food industry with GMO testing, and prenatal diagnostics. For the sake of this webinar, I would like to focus on absolute quantification in the context of HIV infection. Okay, so allow me a brief introduction to where we are in the HIV cure field HIV still accounts for about 2 million infections every year, and when you get infected, HIV attacks and infects your CD4 T cells. Luckily, with current therapy regimens, we can suppress the virus and patients can control HIV infection. However, if the therapy is stopped, we quickly see that the virus rebounds and new infections are there. The reason for this is what we call the latent reservoir. These are dormant infected cells with replication-competent virus, and in order to reach a cure, we really need to tackle this reservoir. A first step to reach this would be to actually be able to accurately measure these infected cells, because we know that only a small fraction of viruses in the body is intact and replication competent. Estimations are that only between 1 and 5% of all viruses in the body are so. This comes because often viruses contain deletions and hypermutations due to our immune system fighting off these infections. So this makes that the assays we use to measure total HIV DNA pick up too much and overestimate true copies of intact virus, as these assays are typically designed in conserved regions of the HIV genome. In this light, digital PCR can offer a solution, and a duplex assay was recently described by Brunner et al. In Nature. This duplex assay is designed to 2 regions of HIV, of which we know are often deleted. So hence, if both the signals are present, the blue and the green, that means that the virus is intact, doesn't harbor a deletion, and is potentially replication competent. Another feature of this assay is that one of these deleted regions we know can be hypermutated due to our host defense. So the authors make a use of a competitive dark probe to actually try to ignore these viruses and only visualize the intact virus. So we performed this experiment as well, and first on a Bio-Rad QX200 system. So here you can see the results of this, where the double positives on the top right show the intact fraction in orange. And then the single blue and single green positives show that there are viruses there with deletions or hypermutated viruses. Well, we were then interested in just how much of these green negative droplets are actually hypermutated, because you cannot tell because there is a dark probe used. Well, as a first experiment, we just repeated this on the Naica platform with similar results, which is good, but then actually added a third color with a Psi5 probe to hit the hypermutated region. And as you can see, by doing so, we can actually visualize how much of the green negatives are carrying hypermutations, which in this case was 3.5%. So in the end, this is really information that remains unknown in the Bio-Rad QX200 system and can be made visible with the Naica platform. And this really shows the added advantage of an additional channel and even going up to triplex or quadruplex assays. A nice feature of the Naica is that also the raw data is made available with each run, which allows further processing if desired. So our next steps with this assay is to measure patient samples and quantify the latent reservoir, even in the context of HIV cure strategies, which examine whether there is a reduction or not of the latent reservoir in the context of a cure. In the final part of this webinar, I will talk about some challenges in the digital PCR field and suggest some future directions. So digital PCR technology is really not only unicorns and rainbows, but also has some challenges. So we at the Ghent University have 8 years experience in our consortium with digital PCR, and was mainly started with the Bio-Rad and recently acquired the Stilla platform. We're dedicated to find data analysis solutions based on our experience with this technology and introduce digital PCR in the clinic. In the recent years, we published over 15 papers on this and constructed 7 data analysis tools to handle digital PCR data. So if we go into the challenges. One of the first things we noticed is that often there are partitions with intermediate fluorescence. In an ideal world, these wouldn't exist, and it would be easy to count the positives and the negatives. But it often is not the case, and these intermediate fluorescent droplets are referred to as rain. We try to address this phenomenon in the following experiment, where we actually have 0 or we introduced 1 or 2 mismatches in the probe binding region. And these you can see in the sections A, B, and C. But if you merge these 3 together in section D, you can actually see that we created rain and that this is a real source of positive droplets. So we show that these need to be included in the analysis, as other tools actually discard these. So this is a very important finding, but this might not be always the case. And the real reason of these intermediate fluorescent droplets or partitions are still under investigation. The second issue was the observation of positive signal in the negative template controls, as you can see here. We then left out forward and reverse primers, and we also observed some of these positive partitions, leading us to the conclusion that this is probably a combination of artifacts and contamination. So I do have to stress that this data is based on our experience with the Naica system, and we still have to explore this on the Naica platform. So as a future direction, I think it would be nice to have additional partition quality control in the form of melting curve analysis or sequencing to actually check whether these amplicons are the desired ones or not. Next to this, digital PCR still has a higher cost per sample than qPCR, so it's still a bit more expensive. So it might be an option to lower the price to really compete with qPCR more. The next thing is that throughput can be an issue, but it really depends on the application you have. But what was most striking for us and our consortium is that there was no real standardized data analysis pipelines or reporting. So, as I explained, we developed several data analysis tools to help out and correct for technical aspects we observe. A first one was the observation of a baseline shift between samples. So if you draw a threshold, this is highly problematic, and it could actually run into the negative fraction of your droplets or partitions. So we have made a method to correct for this. A second observation was, if you multiplex, that the compensation is not always ideal. You would expect that the blue positive cloud would be right on top the negative black one. So by running mono colors, so that means singleplex, you can actually compensate for this and get to the correct alignment of these droplets. The third thing was, and that's a very important one, it's threshold determination. And this is really a key aspect because this determines the positive versus the negatives and, of course, your quantification. We developed a statistical framework for this in a tool called dPCR quant, which is based on the Bio-Rad data, but will now also be available for Stilla and other platforms in the future. So we really try to have a statistical framework that works for threshold determination so that everyone can try to do it in the same way, as we know also from flow cytometry that manual gating or manual thresholding has, of course, its own difficulties. Next to that, we also observed that the error propagation, so the uncertainty on your concentration if you have replicates, is also not easy to calculate, and we offer statistical and mathematical solutions for this in a tool called GLMM analysis, which is also used for copy number variation data analysis and free to use as a Shiny app. Next to that, we also developed tools to perform quality control of your experiment to see if everything was done okay, and to calculate power, how many samples you actually need to see a certain difference. For instance, if you want to reach the 0.1% minor variant, how do you approach your experiment? Finally, we also helped to develop the digital MICI guidelines, which summarizes how to report on digital PCR data. And this again is all to uniform the reporting and to make it reproducible so that everyone can actually redo the analysis of the data you generated. So in my opinion, there are some interesting future directions for this field, which includes the integration of single cell setups, the possibility to get partition retrieval for downstream sequencing or sorting. And as I explained before, it's really a key aspect to address the proper statistics in digital PCR, especially in the multiplexing area where more and more colors are being combined. And that will be an interesting aspect to further evaluate. And finally, which I also already addressed, is the need for additional quality control on your partitions, preferably with a melting curve or the possibility to sequence your amplicons. So the good thing is, for the Stilla platform, you can actually backtrace and look at your partitions. So the picture that is made by the camera is made available, and you could actually evaluate whether there is a false positive because there's kind of contamination with a dust particle or not. You can actually look at that. So that is already one step in a good direction, but having additional QC would be beneficial for digital PCR. So in conclusion, I think we really are experiencing a digital PCR renaissance with an explosion of platforms. Hence, for an end user and for myself, it's an exciting time to learn about all these platforms, explore their advantages and disadvantages, and look for applications that are possible. Finally, I would like to thank following people at the Digital PCR Consortium, people at the HVQ Research Center, Stilla Technologies, and Jim Huggett for the help in making this webinar possible. And of course, for the audience, thank you for your attention. And after this, the floor is open for questions.
Thank you for your presentation, Wim. It is now time for the Q&A. To ask Wim a question, just type it in where it says type your question here and then press submit. Now, first question, Wim, and this one asks, how easy or difficult is it to transfer a qPCR assay to digital PCR?
All right, yeah, thank you for the question. So we have experienced that first hand on. So if you have a fully optimized qPCR assay, it's actually, I wouldn't say plug and play, but it's actually quite easy to transfer to the digital PCR setup. What we did notice is that we often have to play with the primer probe concentration a little bit, and often we need to spike in a little bit more of the probe or the primer to get good separation between positive and negatives. But in the end, if you have a well-optimized qPCR assay, chances are very high that it will also work directly on a digital PCR platform.
Excellent. Thanks, Wim. And our next question asks, what is the dynamic range for digital PCR compared to qPCR?
So yeah, there you have one of the advantages of qPCR, that actually the dynamic range can go up to 10 to 12, even 14 logs. But for digital PCR, it's a little lower. It's more in the range of about 5 to 6 logs. So what we actually then do, for instance, if you have a fully positive sample, that means that you actually overloaded it. Then you have to go back and make a dilution and take that dilution factor into account. So if you compare qPCR and digital PCR on the dynamic range, then you see that qPCR has a bigger one.
Perfect. Thanks, Wim. And our next question asks, how easy is it to perform a digital PCR experiment?
Well, yeah, indeed. So we were well used to using qPCRs, and if we train people or if we got to it ourselves, we were actually amazed by how quickly you adapt to the digital PCR workflow. So meaning that the pipetting and the workflow actually goes quite fluent. And with the first experiment, you actually generate already interesting data. So actually, it's quite astonishing how easy the transfer was from qPCR to digital PCR workflows.
Perfect. Thanks, Wim. And just a quick reminder to our audience, if you'd like to ask Wim a question, all you need to do is type it in where it says type your question here and then press submit. Our next question, Wim, this one asks, if you needed to name one key feature of digital PCR, what would that be?
Well, yeah, there are, of course, some multiple key features, but I think one that first pops to mind is the absolute quantification without the need for a standard curve. It really boosts the reproducibility. Yeah, that is really a key feature. I think Jim Huggett has already had some work on that, proven that, for instance, if you send out samples to 38 labs, measuring the same thing, that everyone gets inside, or almost everyone gets inside the 95% interval of the same concentration. And that's something that is unprecedented. Also with qPCR, you need to do the standard curves, and the error that brings to that, and the correction factors, that is just crazy compared with the ease of just not using that and using direct quantification. So I think the main advantage I would, if I need to name one, I think it's the absolute quantification without a standard curve.
Perfect. Thanks, Wim. And our next question asks whether you can comment about the range of digital PCR platform pricing.
Oh yeah. So of course there are multiple platforms out there, but if you're looking to invest, let's say in the QX200 or a Stilla instrument, I think you quickly need to think about a price range between it's 75, 100 and 10,000 euros. I think that's a price range you should think of. Of course, it's up to you how good your negotiation skills are to get some reductions, but I think that's a fair range of the price that you need to think of. Of course, please contact the platform providers. They will of course give you more information on the pricing.
Perfect. Thanks, Wim. And our next question asks, what is the advantage of array systems over droplet systems?
Well, if you talk about, for instance, the Stilla, the arrayed droplet system, I think the nice thing is there that you actually can go back. So you take a picture, you can go back to your arrayed droplets and see, for instance, if they're a dust particle or not. So you have that information there. Also, for Stilla, for instance, it's a closed system, so meaning that you actually can go back and cycle a little bit more and then visualize again. So for the droplet-based systems, you actually lose your droplets after the readout. So if you have an array-based system in a closed setup, then you can actually also retrieve more easily your droplets. So I think that's a couple of the advantages of having an array-based setup.
Excellent. Thanks, Wim. And again, just a quick reminder to our live audience, if you'd like to ask Wim a question, just type it in where it says type your question here and then press submit. Now, our next question asks, what is the sort of controversy around the reproducibility of digital PCR? Has there been any?
I think that's coming back to the same question, but to the answer I gave before. I think the reproducibility is really shown to be higher than in qPCR. Hence the fact all these different standard curves and so on. I also refer to the work of Jim Huggett. So if you're interested, please try to find those publications. But if you have to put a number on it, I think it depends on which papers you use. But it ranges between 2 and 10-fold higher reproducibility.
Excellent. Thanks, Wim. And our next question asks, how does the digital PCR functionality compare to next generation sequencing? Are there advantages of digital PCR in terms of cost, speed, reliability, or precision, for example?
Yeah, indeed. Yeah, so I think those technologies go side by side, I would say. I think it really depends on what kind of question you wanna answer. I think if you have the digital PCR or qPCR setup, it's more a targeted approach that you have for a more select number of targets. Of course, with NGS you can do target enrichment. You can go much more bulky. So it's actually a different type of application. Of course, the cost is going to be different. The speed to results. I mean, I think the cost of digital PCR will be lower per sample than if you run an NGS. In terms of speed, I think there, you would also favor digital PCR if you really want to have a quick result, for instance, in the clinic. Then I think digital PCR or qPCR is quicker. Also, from a data analysis point of view, it's more straightforward to analyze digital PCR data, although it has its difficulties sometimes, but it's nothing compared to running an NGS bioinformatics pipeline, which will take much more bioinformaticians and people to run it. So indeed, on several aspects, there are some differences between NGS and digital PCR.
Excellent. Thanks, Wim. And our next question asks, what is the price difference between qPCR and digital PCR assays?
So indeed, I mentioned also one of the challenges. I think if you run a digital PCR, it's still a bit more expensive. So if I need to put a number on it, I think digital PCR sample would be ranging around 5-6 euros, whereas a qPCR would go as low as I think 2-3, for instance. So I think it's about a 2-3-fold price difference, I think, per sample. But again, I can maybe a little bit off. So please contact digital PCR platform provider if you wanna know more. But I think that's going to be roughly the price range of running digital PCR per sample.
Excellent. Thanks, Wim. And our next question asks, would it be possible for you to elaborate a little bit further on the output of digital PCR? This individual was wondering what the x-axis represents on the graphic that you showed, and what does the y-axis represent?
Okay, yeah. So typically, if you have digital PCR output data, it's been plotted. The fluorescence is plotted. It's in the y-axis. So the fluorescence amplitude of your partition, meaning what is the fluorescent signal of the partition. And then on the x-axis, it depends on how you do, but that's most of the time it's the index of your partition. So that's that. And if you have, of course, a higher fluorescence, meaning that in that partition the PCR happened and you could detect it, that will show up higher on the plot than partitions that won't have the target and the PCR did not run. So that's why you can distinguish them between high positives and low positives. So that's basically roughly the typical data output you generate or visual data you generate with digital PCR platforms.
Excellent. Thanks, Wim. And our next question asks, is there a possibility to perform duplex single dye TACMAN based assays in digital droplet PCR?
Yeah, that's indeed possible. So instead of using different colors to detect different probes, meaning different targets, you can actually also play with your probe and primer concentrations. So, for instance, you can have a 1-to-3 or a 3-to-1, 4-to-1 ratio between those two assays, and they will actually then show up on a different fluorescent intensity will be higher, so you will have different clouds of positive droplets. So there's a way around having there's a way around of running, for instance, high order multiplexing, where you have more targets than you have colors. So it's mainly playing with primer probe concentrations.
Excellent. Thanks, Wim. And just a quick reminder to our live audience again, if you'd like to ask Wim a question, just type it in where it says type your question here and then press submit. So our next question, Wim, this one asks, are there any oncology applications for digital PCR? So, for example, can digital PCR be used for liquid biopsies, for detecting circulating tumor DNA, for example?
Yeah, for sure. Yeah, I think that's in the field of oncology, it's very well used. I mean, the applications are very good in that field. Liquid biopsies, you have low DNA, you can actually quantify, and also I think two main things there are copy number variations and resistance mutations, for instance. And if you want to pick up minor variants, that has been shown in several papers at the digital PCR, you can actually really spot those 0.01% minor variants, for instance. Next to that, you have also copy number variations, which are quite easily to set up at a digital PCR platform. So yeah, especially with the liquid biopsy field, with liquid biopsies being more important in the future, I would definitely advise, if you're in the field of oncology, to really think about how digital PCR can work for you, especially for detecting those minor variants and copy number variations. Yeah.
Excellent. Thanks, Wim. Our next question asks, is sensitivity of detection better with digital PCR than qPCR? I think you touched upon this in a previous answer.
That's correct. Yeah. So I touched up also during the presentation. So in terms of sensitivity, there's still a little bit of debate there. Let's say it's okay. I will put it like this. If we compared it to a normal qPCR, we saw that the digital PCR was actually a little bit better in picking up, so it was more sensitive. But if you have a semi-nested or a nested qPCR approach, then you actually you can reach an equal sensitivity between digital PCR and your qPCR. This being said, of course, I mean, digital PCR is just a one PCR setup. It still means that you need to set up a nested one where you run two PCRs one by one. So still it is more easy to have the same kind of sensitivity. But I will certainly not make the claim that the digital PCR has really a big advantage over sensitivity concerning a fully optimized nested qPCR. Bear in mind, this is talking about picking up, for instance, HIV sequence of HIV RNA transcripts. Again, if you want to go for instance, minor variant detections, where it's only, for instance, one target in a background of 100 wild types, you will be able to pick it up with digital PCR, and you will not be able to pick it up with qPCR. So the sensitivity is actually it differs on which application you're talking about. So yeah, I will leave it that. I will leave it there. Just know that if you want to pick up a minor variant, you will be able to do that with digital PCR and probably not with qPCR.
Excellent. Thanks, Wim. Our next question is asking whether you have any comments about needing a DNA template for assay optimization, for example, positive control or a negative control. Is that required for every experiment?
I think that also depends on the end user, what we typically do. And if you start optimizing assays, it is a good idea to order a synthetic template, make a dilution curve, and see how your assay is performing. So that is actually something what we're doing in the lab. So that's in terms of that's in terms of using a template. The second thing there is using it as a positive control. I think adding positive and negative controls in your digital PCR is always a good idea. And you can actually use this template in a positive control well, of course. Now for the negative controls, also speaking about with the HIV field in mind, it is important that your negative control resembles your sample as good as possible. So what we try to do, what we do is, if we want to pick up HIV DNA or RNA, for instance, we use DNA or RNA from a healthy donor control, just to have it as similar as possible. Water controls is fine, but it's good if you have a target-free negative control, but that if there's still DNA or cDNA in there. So I think that's a bit the answer there concerning templates.
Perfect. Thanks, Wim. Our next question asks, can kits that are generally used to validate qPCR assays be used to validate digital PCR assays?
Well, I'm not sure what is really meant by kits. If you use a validation for a qPCR or a digital PCR, you use probably reference material. And again, it's the PCR is the common ground here. So any reference material you would use for a qPCR, you can actually also use for digital PCR. You just have to watch out then how much input you use of your reference material per sample. If you of course overspike your digital PCR with reference material, you will not get a good answer. So I'm not sure if that answer really fits the question. As I don't really know what was meant with a kit, but if it goes for reference or standard material, of course this is indeed transferable to a digital PCR.
Excellent. Thanks, Wim. Next question asks, is digital PCR more sensitive to primer and probe design than qPCR? So, for example, would primer dimers and or hairpins be more critical?
Well, if we design assays for digital PCR, we try to abide by the rules that you would have for qPCR. So just make that those things don't really happen that much, and try to avoid all primer dimers and so on, and self-annealing. So I would just use the same general rules that you have for qPCR and also for digital PCR. What I can say, if for instance, if there are primer dimers or there is some inhibition, that in your digital PCR you have a little bit more flexibility, a little bit more, it's a little bit more refractory to it. Meaning that, if it happens, the target still will be amplified as well. And if that's already a little bit high enough, it doesn't have to fly entirely, you will still get some separation between your negatives and your positives. So I think general primer and probe rules apply, and maybe the digital PCR is a little bit more flexible if it gets inhibited some way.
Excellent. Thanks, Wim. Our next question asks, how easy is it to use the Stilla data analysis tool compared to the QuantiFost?
Yeah, that's always tricky to answer, right? Let me put it like this: we have only shortly acquired the Stilla instruments, so we don't have a lot of experience yet with the data analysis interface. But what I can say is that it's quite intuitive, and I quite like it. It goes fluent. You can visualize 2D, 3D. So it's actually quite quick and quite nice. This being said, I think the QuantiFost software, which will be updated also next year with a new version, will have its benefits, and it will also go quickly. So I think from a data analysis point of software, I think with the new update that Bio-Rad will do, it will be also fine. The thing that maybe you can say for the Crystal Miner is that the openness in data sharing is maybe a little bit higher, and the export functions are a little bit more elaborate than if you would compare it to the QuantiFost. But in the end, it's software designed to work with their respective tools. So I won't really pick sides here. Still a bit more in the middle ground.
Excellent. Thanks, Wim. Our next question asks, to generate linear regression curve by digital PCR, must one use a standard plasmid, or is it possible to use genomic DNA?
Could you quickly repeat the beginning of your question, Jason? I wasn't.
Yeah, so this individual is asking, so to generate the linear regression curve, do you need to use a standard plasmid, or is it possible just to use genomic DNA?
Oh yeah, you can use both. It's no problem as long as you make the dilution curve as good as possible. So it's not really a preference. You can use genomic DNA, you can use a synthetic oligo, you can use plasmids. It's just where your target is. You can use that. So what I might or could add is if you use genomic DNA, we often perform a restriction digestion before we run digital PCR, because it's been shown that it improves your homogenization and your partitioning. So that's something I can advise. Is look into restriction digestion, and of course, if you do that, please don't select an enzyme that cuts inside your amplicon, of course. But besides that, it's you can use whatever you want as long as the target is present.
Excellent, thanks, Wim. How reliable is digital PCR for quantifying disease burden? For example, can one reliably measure HIV viral load or tumor burden?
Yeah, good question there. I think what we see in our reference laboratory is that we have the well-established qPCR platform that is running well. So coming back to the dynamic range, if you have a patient, for instance, with a very high viral load, you will be able to have it, to pick that up in one go. If you overload your digital PCR, you have to do a new dilution and rerun it. So that could be a little bit more tricky. On the other side, that's an RNA-based measurement. I think for RNA and the reverse transcription applications in digital PCR, we're trying to do it, but what we actually focus on is more DNA based, because with the RT's one-step kits, for instance, we had some experience that was not really generating very good results. Again, we also heard that there is a new version of the RT one-step kit, so which has been apparently will work better, but we're not able to test that yet. So digital PCR for viral load testing and HIV. I think that with the established methods we have now, it's okay, but of course it could have a benefit and no standard curve and so on and so on. But I think it will be a very difficult application to because now it's really well established for the qPCR. So that's a more tricky one, I would say.
Excellent, thanks, Wim. Next question asks, why is the copy number assessment drops? Let me start that one again. Why does the copy number assessment drop with amplicon length?
Well, I don't have it on the top of my head because we're not running a lot of copy number assays. But if your amplicon is shorter, you get preferential. Your PCR will go much more quick. So it will be much quicker than if your amplicon is longer. That's the only thing I can just think of on the top of my head. But I'm more than happy to answer that more in depth offline. Given the fact that if I have the contact information of that individual.
Yes, absolutely. When we can pass that on, and you can sort of elaborate further with that individual. Our next question asks, how high should the RFU difference be between positive and negative droplets?
Yeah, so that's always. So there's no clear answer. Let me start with that. From a theoretical point of view and data analysis point of view, if you can see the two populations, you can perform a quantification. This being said, there's not really like a strict are a few, like you have to have a 2,000 or 4,000 or 5,000 difference. There I would advise to run a gradient PCR, check your on different annealing temperatures, and see if you can actually increase the separation by running this gradient. And there you can actually see if running a higher or lower temperature will actually make your spread bigger or not. So it's not like a fixed quality control value out there yet. I think it would be nice that the digital PCR community would think about that and get some kind of measurements to perform a quality control on that aspect. So yeah, I think I will leave that there.
Excellent. Thanks, Wim. Well, I'm afraid that is all we have time for today. Before you leave us, we would really appreciate if you could fill in a very quick survey. I would now like to thank Dr. Wim Tripstein for his presentation and for answering your questions. I would also like to thank the webcast sponsors Stilla Technologies, and of course you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand at nature.com webcasts. Thanks for watching, and I hope you'll join us again soon.